Okay, so today what I'll do is I'll give you a little bit of introduction about our company, what we do. We'll talk a little bit about biologic degradation and the sources, and I'll give you a specific example that maybe you haven't considered about our packaging and what our technology brings to the game. If you just consider glass alone, or maybe you're considering even plastic alone. We're a hybrid solution, as you'll learn a little bit later. But we consider ourselves a safe solution, especially when it comes to various sterilization technologies, and I'll use that as a bit of a case study for you. So who are we? SO2 Medical Products, or SO2 Material Science. We do have a long history of various specialty products, and while we were formed in 2009, our owner, Bobby Abrams, has formed many businesses and a lot of innovative products over the course of about 50 years. So you can see everything from the first milk jug with integrated handle to various vials with integrated flip-top caps. We make, if you've ever seen the mini M&Ms, those are made actually in Auburn, Alabama, and then a whole host of other products, as you can see. Now, up to about 2009, when we were formed, there's a few other products that we've launched. One for a malaria vaccine in a vial for cryogenic storage. Also for an ophthalmic syringe, a transfer syringe for an ophthalmic application, and then finally a blood tube that contains a blood preservative for cell-free DNA and RNA. So just to name a few things along the way. So what does our businesses consist of? They're all driven by certainly our customer needs. The one I'll be focusing on today will be our pharma and biotech solutions, which focuses on primary packaging for biologics and biosimilars. It also includes things like cell therapies, cryogenic vials, as well as lyophilized drugs. We also have two others, our genetics and diagnostic solutions. I talked a little bit about our blood packaging solutions, our blood tube packaging. And then also the final is our performance products, which covers a lot of different areas, mainly focused on safe products and leachable-free products for like baby bottles, coffee capsules, and the like. Okay, so what makes us different? Well, if you look at all of the various industry problems, which I know you all are familiar with, we address all of these. And if you think about just glass alone or plastic alone, individually, they don't address everything, okay? So you have to really think about your packaging solution. What makes us unique is we're a hybrid material. We basically take the best of glass and the best of plastic and integrate it into one solution without the shortcomings of either. So this gives you just a short list of those problems that we've solved. And one of the things I'll be focusing on today is around the stability and shelf life. So you put a drug product into a primary package that's been sterilized, and I'm going to talk to you a little bit about radiation sterilization technology in particular and why our technology is advantageous. So let's talk first about degradation of drug products, specifically the biologics. Of course, there's a number of different areas where one would have to consider possible sources, all the way from manufacturing and formulating to packaging and storage, even transport, and then finally the delivery of the drug itself. And I've just listed many of the things I'm sure you guys are all familiar with. I think Christian has touched on those this morning as well. If we dig a little deeper, what sort of pathways of biologic degradation could you conceive of? Well, there's many physical as well as chemical instabilities that can occur. On the physical side, of course, there's colloidal instabilities that can be formed. Conformational changes, in this case a monoclonal antibody that can lead to denaturing of the biologic itself. And then a whole host of chemical pathways. I'm just listing some of those. Oxidation being probably one of the most well understood, but maybe one of the most challenging to address in formulation science. Probably because there's just so many different pathways a drug could degrade. The diversity of drugs are very different as well. And I will focus more on oxidation today when it comes to sterilization. So ultimately, what does this all mean? So regardless of how the drug degrades, ultimately you'll get the drug to unfold, aggregate, and then it's injected into the patient. And ultimately, there's possibly an immune response. It could be a rash, it could be anaphylaxis, it could be death. So this is certainly a risk, and what I'll show you a little later is how our package is considered a safer solution when it comes to things like this. And I'm going to, again, use the radiation sterilization technology as a case study. So there's a very good series of papers published by Tomo. And basically what they said here, this in particular was by Nakamura and Kiminami, they both basically had showed if you sterilize a plastic container, in this case a cyclic olefin container, and you sterilize it with either E-beam or gamma, and then you package your drug in it, you can possibly see oxidation of the drug. Now, in this case, they used erythropoietin, and you can clearly see the oxidation occurring over time over the course of 12 weeks. And then they compare that directly to steam and EO, and of course you don't see any oxidation of the drug. So basically what they're saying is, don't sterilize our product, our plain ordinary COP container with either gamma or E-beam, okay? And the studies, it was very well done. In fact, we actually duplicated some of the results ourselves in comparing with our own technology. And I'll show you the reasons why we have an advantage when it comes to this. So if you're going to use ordinary plastic, use steam and EO. And there's other methods as well, but not radiation. Okay, so why would we be any better? So this is essentially a bit of a snapshot a simple snapshot of our technology. You can see, obviously, we coat and produce many different container sizes. We call this our Cyoplast primary containers. Consists of a polymer and then a coating on the inside. Okay, and I'll tell you a little bit more about how we manufacture this in the later slides. So it starts with molding. And basically what we do is we injection mold a preform out of this COP resin. We stretch and blow it into a final mold, and you get either a vial or, in this case, it's injection stretchable mode, so this is specifically for vials. In the case of syringes and cartridges, you'd be injection molding it. And then you apply the coating. So this is a process known as plasma-enhanced chemical vapor deposition. Essentially what we do is we treat the part as a mini chamber. So we remove the air from it using a pump. We introduce a gas mixture to the interior. You apply a voltage across the part. And as soon as you apply the voltage across the part, it's like flipping on a light bulb. You get this glow on the inside. As soon as that happens, you're getting deposition of a coating. And it's a glass-like coating, as you'll see a little bit later. But irrespective, you can coat very extreme aspect ratios, very odd geometries with it. We have a lot of patents on the technology that covers a lot of this on the process side, on the inspection side. And you get a coating that's highly inert, won't interact with the drug product, and it's consistent. The entire interior is coated uniformly. So there's a little bit about the chemistry. I won't dig into this too much, but the key here, because I get a lot of questions about, well, so you got to clean it, you got to activate it, you have to do anything to the part before you coat. And the answer to that question is no. It's a completely dry process, doesn't require any solvents, any cleaning whatsoever. We coat directly on the polymer. And this gives you an idea of the sort of chemistry. This is actually a liquid at room temperature. We deliver this as a vapor, mix it with oxygen at very low pressure. And then, of course, we form the plasma to produce a coating. This is a silica-based coating. In this case, it's got a little bit of carbon in there. And you get residual gases like water and CO2 that go through the vacuum system. So the coating architecture and chemistry, this is basically a snapshot of that. You can see we have a 3-layer structure. The chemistry over here you can see in a visual sense, in an atomic visual sense. So you can clearly see we do have a pure silica layer in the middle. And then the 2 other layers have a silica layer, but also has carbon built into it. The presence of the carbon is really essential for not only binding the entire stack to the polymer through these very covalent bonds with the polymer backbone, and then also for protecting the silica layer. Without any protection, the silica would dissolve like any other glass. Okay, so you have to protect it. And it's maybe hard to tell, but this entire stack is about half a micron. So you think of a human hair as maybe 40 microns. So this is half a micron in total thickness. And one of the individual layers is actually about 50 nanometers. So individual layers are actually even thinner than that. Okay, so very, very thin overall, covering the interior surface. So as I said before, it's chemically inert, it's hydrophobic by nature, covalently bound, and then also has great barrier characteristics. Not only does it prevent any leachables coming from the polymer, but it also blocks things like oxygen, ETO, other gases. We measured the oxygen ingress through a method we developed internally. And basically, this just shows you a little bit of the math and the difference between a coated container and an uncoated container. You're basically measuring the partial pressure of oxygen in the container over time. You can see how very little ingress occurs in the PCVD coated container versus an uncoated. We basically effectively reduced or increased the barrier, reduced the permeability by about 33 times in this case. In some other cases, it's 60 to 100 times, depending on the container geometry and what we want to achieve. So this is just a little bit of a perspective on, so how do you compare it to an uncoated container and to a glass container? And then what's the contribution of oxygen coming in through the closure and through the wall? So here's an uncoated container. You can see obviously a lot coming through the wall versus the stopper. And then here's us compared to a glass vial. There's a slight difference, obviously, in the permeability through the wall, but by comparison, it's very, very similar. Total, if you look at the entire package, including the closure. I've got examples of syringes, the same sort of relationship exists. Okay, so let's come back to the whole area of radiation sterilization using either E-beam or gamma. What actually is going on when you sterilize a container to make it ready to fill to put a drug into it? Okay, so for plastic, what ends up happening, consider the wall of a COP container. You have air on both sides. You have some dissolved oxygen in the polymer itself. You hit it with, this actually should be gamma. That's a mistake. It says E-beam. But you hit it with gamma, and you form what's called these free radicals in the polymer. Okay? And what ends up happening is the free radicals end up getting scavenged by oxygen in the polymer. And then, of course, you get oxygen diffusing in and scavenging those free radicals. And you can see that happening over here. Okay, this is for an ordinary, plain, uncoated COP container. So what happens when you have a coating? Well, the coating's a barrier, so it doesn't allow oxygen from coming through the barrier coating into the polymer to scavenge these radicals. You only have oxygen coming in from one side. And we've actually are looking at the number of free radicals that are in the polymer itself, and you can see the difference in the decay rate because of the barrier coating. And why is that important? Because when you then put the drug product in, then you're basically preventing any of those oxidative species getting into the package and oxidizing the drug. And I'll show you some data a little bit later on how we actually accomplish that. So just to set up those experiments, we ran a study at the University of Chicago in the US. And we looked at 2 different drug formulations. We chose these in particular because they're, in general, considered more oxidatively unstable than most other drugs. Teriparatide is a peptide. It has 2 methionine amino acids in its backbone that make it susceptible to oxidation. So very good candidate. And then erythropoietin has an amino acid that also is oxidatively unstable within its interior, but it's still accessible. It can still be oxidized. And what we did was, normally you would find an antioxidant in the formulation. We took that out just so we're looking at the drugs by themselves. And then, of course, we've already sterilized the container. We put the drug in it. And then we ran these oxidative measurements. One for teriparatide using reverse phase high-performance liquid chromatography, which is a pretty standard method. And then for the erythropoietin, since Nakamura had used this same drug, we basically mirrored the exact same analytical technique he used to look at oxidation of that drug. One other thing is, in this case, we're using vials, so we actually, the surface area to volume ratio was, we tried to mimic that of a syringe. We actually are duplicating these results in syringes, but just to mirror that for a syringe. Okay, so these are results. What you see here basically is percent oxidation of teriparatide over the course of 25 weeks. And you can clearly see gamma and E-beam sterilization of a COP container containing teriparatide. You can see the oxidation occurring, not surprising. And then in this range, you're seeing obviously the gamma sterilized glass in our container, and then an unsterilized COP. So you can see a little bit of chatter there. Of course, you can imagine when you're sterilizing a batch or a large batch of containers, there's certainly variations in dose throughout the batch. We attribute some of that variation to things of that nature. And then erythropoietin, in this case, you can see percent oxidation of gamma and E-beam sterilized vials in COP. So COP alone, about 12%, so about double that of what you'd find in a glass vial. And then compare that to our vials, E-beam and gamma sterilized, and again, you can see about 6%, so very similar to a glass vial by comparison. And again, this was after 25 weeks. So finally, hopefully you understand a little bit more about one of the advantages. We obviously have a lot of advantages, but I wanted to use this as an example to show you if you're indeed considering gamma or E-beam sterilization of a drug product that does have instabilities with regard to oxidation, this is a great choice. And particularly if you're concerned with possible aggregate formation as a result of oxidation, which could potentially lead to immune response. So that concludes my talk. Thank you, and if any questions, please. Okay, Kristen.
Yeah, okay. Thank you very much for this nice talk and showing these good data related to oxygen permeation. Are there still any questions from the audience?
Crystal clear?
No.
Oh, sorry, yes.
So usually from glass we know a kind of a discoloration reaction when glass is in contact with E-beam or gamma.
Yeah.
Thanks. I'm not sure if this layer is too thin to even see that. Can you comment on that, please?
Yeah, so certainly the polymer would be affected by that, but it depends on dose. So as you go higher in dose, you'll see discoloration of the polymer, not the coating. I mean, it's again half a micron in total thickness, so that's not going to affect anything. But depending on dose, it's completely reversible for the polymer. You'll see some discoloration, and then it goes right back to clear again. If you go too high a dose, it can be irreversible. But we basically sterilize at a very low dose, and it's perfectly fine. So typically in the range of about 15 to 20 kilograms is typical, and you won't see any color change.
I have one question.
Sure.
You mentioned during your talk that the thin of the layer is really, really small.
Yes.
And from my perspective, we see the data are really, really good. Yeah. But how you can, ensure that the layer is, over the whole inner surface of the vial or the syringe, and how you can detect it during in-process controls, for instance, or how you ensure that the layer is really good on the surface.
That's a great question. Of course, we didn't go into our manufacturing or our inspection technologies, but so we do have a lot of inline inspection on the line that not only manages the process parameters, but also actually looks at the coating as well as the plasma. So we have some vision systems that look at the plasma light, make sure that that's uniform. We also use inline infrared spectroscopy in some cases, which actually can detect the coating and ensure that it's there and that you have all the layers there as well. And then there's another inspection that actually looks at barrier, believe it or not. So we've actually pressurized vials with CO2 and then pull the CO2 back out. And you're basically looking at desorption of CO2, and that correlates to the barrier. So we've got a number of systems in line with our manufacturing to ensure the coating's there and it performs as we would expect it to.
Okay, thank you very much. Well, this could be a critical quality attribute in the future, so that it could be that we have, when we as a pharmaceutical company implemented your vials, we have to test if these surface really closed and there, that's a question in the end.
So I value your comment because it is so critical, and because of it, we probably spent as much time developing the inspection technology as we did the the actual container itself with a coating, because it is so critical. Yeah.
Okay, thank you very much.
All right, thank you.